Welcome to this video lesson on biochemistry topic, metabolism of purines and pyrimidine nucleotides. This topic is divided in two parts and this corresponds to second part. This will have a focus on biosynthesis and regulation of pyrimidine nucleotides, catabolism of pyrimidine nucleotides, and catabolism of purine nucleotides. Biosynthetic pathways of purine and pyrimidine nucleotides are conserved among all organisms. Notably, these two pathways do not overlap with each other, but essentially begins with metabolic precursors such as amino acids, bicarbonate, a coenzyme called formilyl tetrahydrofolate, methylene tetrahydrofolate. Phosphoribosyl pyrophosphate gives a start to purine biosynthesis, where a ribose phosphate sugar serves as a foundation or a scaffold on which purine ring is synthesized. On the other hand, in pyrimidine nucleotide synthesis, pyrimidine ring is synthesized first and then it is attached on ribose phosphate sugar that is donated from phosphoribosyl pyrophosphate. In both these cases, non-canonical nucleotides are synthesized, and these nucleotides serves as precursors for canonical nucleotides. Let's view pyrimidine biosynthesis. A radioisotopic labeling experiment has shown ring atoms of pyrimidine have been acquired from precursor molecules. Carbon 4, carbon 5, 6 and nitrogen 1 is acquired from aspartate. Carbon 2 is acquired from bicarbonate. Nitrogen 3 is acquired from glutamate. Pyrimidine nucleotide synthesis pathway involves 6-step reactions that eventually gives uridine monophosphate as a pyrimidine nucleotide synthesized. Orotate, a non-canonical pyrimidine ring, is synthesized first and then attached on ribose phosphate sugar, which is donated by phosphoribosyl pyrophosphate, and that gives a non-canonical nucleotide called orotidine monophosphate or orotidylate. Orotidine monophosphate is then decarboxylated, and that gives a canonical nucleotide called uridine monophosphate. A uridine monophosphate is further phosphorylated to uridine diphosphate, uridine triphosphate, and uridine triphosphate through amination gives cytidine triphosphate. This is a list of products, and in parentheses the effector enzyme have been put. The first product of pyrimidine biosynthesis is carbamyl phosphate. Second one is carbamyl aspartate. Next is dihydroorotate. Next is orotate. Next is orotidine monophosphate or orotidylate. And the final one is uridine monophosphate. Let's have an overview on pyrimidine biosynthesis. The pathway begins with incorporation of bicarbonate, couple of ATPs, amino acid called glutamine. All they incorporates and gives a product called carbamoyl phosphate. And this step is mediated by enzyme called carbamoyl phosphate synthetase 2. This is a cytosolic enzyme that gives a product called carbamoyl phosphate. In the next step, aspartate participate into the reaction and that condenses with carbamoyl phosphate and gives a product called carbamoyl aspartate. The step is mediated by aspartate transcarbamylase. A next step that carbamoyl aspartate is converted to dihydroorotate through intermolecular condensation or dehydration that gives a pyrimidine ring synthesized as a dihydroorotate. The step is mediated by dihydroorotase. In the next step, dihydroorotate is oxidized to give a non-standard pyrimidine ring called orotate. The step is mediated by dihydroorotate dehydrogenase. This is a mitochondrially located enzyme that oxidizes dihydroorotate and converts it into orotate. In the next step, orotate is attached or coupled on ribose phosphate sugar that is donated by phosphoribosyl pyrophosphate, and that gives a non-canonical nucleotide called orotidine monophosphate. The step is mediated by OMP decarboxylase. And in the next step, uridine monophosphate, a canonical nucleotide, is synthesized through decarboxylation reaction, and the step is mediated by OMP decarboxylase, and that gives a nucleotide, pyrimidine nucleotide, uridine monophosphate. This nucleotide is further phosphorylated and that gives uridine diphosphate. The enzyme that acts on it is UMP kinase. And next reaction, uridine diphosphate is converted to or further phosphorylated to uridine triphosphate. And the enzyme is nucleoside diphosphate kinase. Uridine triphosphate is converted into cytidine triphosphate through an amination process where glutamine is converted to glutamate, and the amide group is donated to uridine triphosphate, and that gives you cytidine triphosphate. The enzyme, the step is mediated by cytidine phosphate, triphosphate synthetase. This is a structural depiction of pyrimidine biosynthesis. The X-ray structures of various enzymes, these are the respective enzymes, and their respective X-ray structure have been shown. The pathway essentially begins with incorporation of two molecules of ATP, bicarbonate, glutamine, and water molecule. In the first place, bicarbonate is activated as a carboxyphosphate by getting one phosphate group from ATP. The first ATP donates one phosphate group to bicarbonate, and that gives you a carboxyphosphate. Further, carboxyphosphate is turned into carbamate. A glutamine amide group is acting on carboxyphosphate, and that displaces the phosphate group and form a carbamate. The carbamate is further phosphorylated with the second ATP phosphate group, and that gives carbamoyl phosphate. This is an active intermediate formed in a first step of pyrimidine biosynthesis. In the next step, carbamoyl phosphate is condensed with aspartate, and that gives carbamoyl aspartate. The step is mediated by aspartate transcarbamylase. In the next step, carbamoyl aspartate is condensed It's intermolecular condensation or dehydration that gives a ring form. It's a pyrimidine ring form that is called dihydroorotate, and the enzyme which mediates this step is called dihydroorotase. Thus, formed pyrimidine ring is further oxidized to a non-standard pyrimidine ring called orotate. The step is mediated by mitochondrially located enzyme called dihydroorotate dehydrogenase. This is located in the outer surface of inner membrane of mitochondria. The oxidation step gives orotate, a pyrimidine ring form, and this ring is further attached on ribose phosphate sugar in a beta configuration. The step is mediated by orotate phosphoribosyl transferase, where phosphoribosyl pyrophosphate donates ribose phosphate sugar, and the sugar on which the orotate ring is coupled, and this forms a non-canonical pyrimidine nucleotide called orotidine monophosphate. In the next step, orotidine monophosphate is decarboxylated and uridine monophosphate is synthesized. The step is mediated by OMP decarboxylase. UMP as a canonical pyrimidine nucleotide serves as a precursor to synthesize cytidine triphosphate and other pyrimidine nucleotide called deoxythymidine monophosphate. In a prokaryote or in a bacteria, pyrimidine biosynthesis takes place as a 6 discrete reactions mediated by 6 discrete enzymes or distinct proteins, and each step is independently catalyzed, and that overall gives a synthesis of uridine monophosphate. Whereas in case of animals or in mammals, all 6 enzymatic reactions are distributed among 3 proteins, 2 of which are multi-enzyme complex or multifunctional polypeptides. Our initial 3 steps are mediated by carbamyl phosphate synthetase, aspartate transcarbamylase, and dihydroorotase. These 3 enzymatic activities are located on single cytosolic trifunctional polypeptide that act as a multi-enzyme complex and having 3 different catalytic sites for 3 different enzymes. This is a 210 kilodalton cytosolic protein polypeptide that act as a multi-enzyme complex and mediates these initial 3 steps. A first step is mediated by a separate enzyme called dihydroorotate dehydrogenase. This is a mitochondrially located enzyme, and that turns dihydroorotate into orotate, or that oxidizes dihydroorotate into orotate. Step 5 and 6 are mediated by orotate phosphoribosyl transferase and OMP decarboxylase. These 2 catalytic activities are residing on a single cytosolic polypeptide, collectively known as a UMP synthase, and that eventually gives uridine monophosphate as a precursor to synthesize other pyrimidine nucleotides. Having this activity on a single polypeptide confers some advantages to an organism to synthesize purine or pyrimidines. A substrate that is acted by first catalytic site turns into product, and that product is channelized to another catalytic site, and that is turned into product, and that product is further channelized to another catalytic site. And overall, this shortens the time of one product to move to another catalytic site or another enzyme and serve as a substrate. So it shortens the time in first place, and then it also prevents a reactive intermediate or product to get diffused into cytosolic environment and get deactivated or being acted by another enzyme. So it prevents that diffusion and deactivation of intermediate. This seems to be a evolutionary development, and that confers advantages to biosynthetic pathways in metabolism. Uridine monophosphate that gives uridine triphosphate and cytosine triphosphate synthesis through UMP is converted to uridine diphosphate, and that is further phosphorylated to uridine triphosphate through a couple of enzymes called nucleoside monophosphate kinase and nucleoside diphosphate kinase respectively. Uridine triphosphate is then converted to cytidine triphosphate through an amination process where glutamine transfer amide group to uridine triphosphate, and that is turned into cytidine triphosphate. The step is mediated by CTP synthetase, and this is a X-ray structure of magnesium ion bound CTP synthetase. This is taken further to synthesize another pyrimidine nucleotide. Cytidine diphosphate or uridine diphosphates are converted into deoxycytidine diphosphate or deoxyuridine diphosphate through an action of ribonucleotide reductase that turns them into deoxycytidine diphosphate and deoxyuridine diphosphate respectively. These are further phosphorylated to have deoxycytidine triphosphate and deoxyuridine triphosphate through action of nucleoside diphosphate kinase. Deoxycytidine triphosphate is deaminated by deaminase enzyme, and that is turned into deoxyuridine triphosphate. Deoxyuridine triphosphate is further converted to dephosphorylated to deoxyuridine monophosphate through an action of deoxyuridine triphosphate pyrophosphatase. This action of deoxyuridine triphosphate pyrophosphatase ensures that deoxyuridine triphosphate is not incorporated in a DNA synthesis. Once deoxyuridine monophosphate is formed, this is further taken to convert into deoxythymidine phosphate, and that is mediated by an enzyme called thymidylate synthase. This requires a coenzyme called methylene tetrahydrofolate, and this is a depiction of regeneration of methylene tetrahydrofolate along with potential targets for anti-cancer therapy and antibacterial therapy. This is the thymidylate synthase enzyme which turns deoxyuridine monophosphate into deoxythymidine monophosphate. During this step, it requires a coenzyme called methylene tetrahydrofolate. This gets oxidized to dihydrofolate. Methylene tetrahydrofolate act as a donor of methyl group or a donor of carbon 1 unit, and that unit is utilized to methylate deoxyuridine monophosphate to deoxythymidine monophosphate. And during this step, methylene tetrahydrofolate is oxidized to dihydrofolate, and dihydrofolate is further regenerated as a tetrahydrofolate through an enzyme called dihydrofolate reductase. This requires a reduced form of NAD called NADPH, which get oxidized and turns into NADP, and that gives hydrogen ions to reduce dihydrofolate into tetrahydrofolate. Tetrahydrofolate is further acquires a methylene group on it, and that regenerate methylene tetrahydrofolate through an action of an enzyme called serine hydroxymethyl transferase, which acts on serine and transfer the hydroxymethylene group of serine to tetrahydrofolate and turns the same into glycine. And the regenerated methylene tetrahydrofolate is further ready to take a next round of methylation of deoxyuridine monophosphate into deoxythymidine monophosphate through an enzymatic action called thymidylate synthase. Here a uracil analog called 5-fluorouracil that is turned into fluorodeoxyuryl monophosphate, a nucleotide that irreversibly binds to enzyme thymidylate synthase, and that blocks the action of thymidylate synthase to turn deoxyuridine monophosphate into thymidine monophosphate. And this a permanent or irreversible blockage of thymidylate synthase stop the conversion of deoxyuridine monophosphate into thymidine monophosphate, and this further hampers the DNA synthesis process of rapidly dividing cancer cells. So this enzyme acts as a potential target for drugs to act as to provide anti-cancer therapy, and prodeoxyuryl monophosphate as a uracil analog is used to as an anti-cancer drug. Similarly, methotrexate is also more or less irreversibly binds to next enzyme, dihydrofolate reductase, and blocks the conversion of dihydrofolate into tetrahydrofolate. Methotrexate, another analog called aminopterin, both are having 1,000-fold affinity towards dihydrofolate reductase than dihydrofolate, and they irreversibly blocks the catalytic site of enzyme and thus blocks the synthesis reduction of dihydrofolate into tetrahydrofolate. Similarly, there is a trimethoprim which also acts as an antibacterial agent, and that blocks the dihydrofolate reductase activity of bacteria. This is the X-ray structure of thymidylate synthase, which shows deoxyuridine monophosphate in blue and inhibitor analog in green. Similarly, this is the X-ray structure of dihydrofolate reductase. The molecule in a golden color is a coenzyme, and a molecule in a red color is methotrexate. Let's view a regulation of pyrimidine biosynthesis. A regulation of pyrimidine biosynthesis in animals or in mammals is mediated by an enzyme called carbaryl phosphate synthetase 2. Carbaryl phosphate synthetase 2 mediated step is considered as a committed step of pyrimidine biosynthesis. This also considered as a regulatory step of pyrimidine biosynthesis. The enzyme is regulated through feedback activation and feedback inhibition mechanism. A purine nucleotide called ATP and phosphoribosyl pyrophosphate act as activators, binds on allosteric site and activates the enzyme to synthesize pyrimidine nucleotides. Whereas synthesis of uridine diphosphate and uridine triphosphate inhibit the enzyme through a feedback regulatory loop, and they act on inhibitory allosteric site present on enzyme, and they inhibit the synthesis of uridine monophosphate and consequently the synthesis that gives uridine diphosphate and uridine triphosphate. Altogether, a purine nucleotide which act as an activator in this synthesis promote the synthesis of pyrimidine nucleotides, and when pyrimidine nucleotides are synthesized to enough amount, they inhibit the synthesis, further synthesis of pyrimidine nucleotides, and through feedback inhibition, they inhibit the enzyme and shut down the synthesis of pyrimidines. A second level control also takes place at another enzyme called orotidine monophosphate decarboxylase. Uridine monophosphate and to a lesser extent cytidine monophosphate act as a competitive inhibitors and limits their own synthesis through inhibition of orotidine monophosphate decarboxylase enzyme. And this is a structural depiction of carbamyl phosphate synthase regulation. ATP act as an activator of the first step mediated by carbamyl phosphate synthase 2 enzyme. Similarly, phosphoribosyl pyrophosphate also act as activator to induce the synthesis of pyrimidine nucleotides, whereas inhibition of this pathway takes place through uridine diphosphate and uridine triphosphate. They act on an inhibitory site of enzyme and block or shut down the pathway through allosteric inhibition of this enzyme. Let's have a view on pyrimidine catabolism. Pyrimidine catabolism results into degradation of pyrimidine nucleotides and pyrimidine prebases to reminiscence of precursor molecules and carbon dioxide and ammonia. A non-specific phosphatases or nucleotides acts on nucleotides and converts them into nucleosides. An enzyme called pyrimidine deaminase deaminate the cytidine or deoxycytidine and convert them into uridine and deoxyuridine. Further, an enzyme called uridine phosphorylase dephosphorylate or hydrolyze uridine glycosidic bond of nucleoside called uridine and deoxyuridine or thymidine and convert them into a free base called uracil and thymine. Uracil and thymine are further degraded through a common reactions mediated by common enzymes, and they are further degraded to, in case of uracil is degraded to carbon dioxide, ammonia and beta-alanine. Beta-alanine is further converted into malonyl coenzyme A and eventually it is converted to acetyl coenzyme A. Thymine is degraded to carbon dioxide, ammonia and beta-amino isobutyrate that is further converted to methyl malonyl coenzyme A and succinyl coenzyme A. And this is a structural depiction of pyrimidine catabolism. This is a nucleotide called cytidine monophosphate, and this is uridine monophosphate and thymidine monophosphate. They are hydrolyzed by enzyme called nucleotides and converted into a nucleoside called cytidine in case of nucleotide called cytidine monophosphate, and uridine or thymidine in case of a nucleotide uridine monophosphate or thymidine monophosphate. A nucleoside cytidine monophosphate is further deaminated and converted into uridine. The enzyme which acts on cytidine and deaminate the cytidine is cytidine deaminase. Uridine and thymidine are further hydrolyzed to pyrimidine base called uracil and thymine, and a liberated ribose sugar is further incorporated with inorganic phosphate and converted into ribose 1-phosphate, and that can be further taken back or participate into metabolic pathways. The step is mediated by uridine phosphorylase, and thus generated free bases called uracil and thymine are further converted to dihydrouracil. In case of uracil, this is further converted to dihydrouracil, and in case of thymine, this is converted into dihydrothymine. The enzyme which reduces these free bases to dihydrouracil and dihydrothymine is dihydrouracil dehydrogenase. The reduced pyrimidine ring is further hydrolyzed to have beta-uridopropionate. Dihydrouracil is converted to beta-uridopropionate, and dihydrothymine is converted to beta-uridoisobutyrate. The step is mediated by hydropyrimidine hydratase. Thus formed beta-uridopropionate and beta-uridoisobutyrates are further hydrolyzed, and carbon dioxide is released, ammonia is released, and that eventually gives beta-alanine and beta-aminoisobutyrate. Uridopropionate is turned into beta-alanine, and uridoisobutyrate is turned into aminoisobutyrate, and they are further converted into malonyl coenzyme A. Beta-alanine is converted to malonyl coenzyme A, and beta-aminoisobutyrate is converted into methylmalonyl coenzyme A. And these coenzymes participate into fatty acid synthesis pathway, and they are eventually converted into succinyl coenzyme A and acetyl coenzyme A, and they are further fed or fueled into TCA cycle. Let's view a purine catabolism. Purine catabolism eventually gives a degraded product called uric acid. Purine nucleotides or pre-purine bases are oxidized to give a degraded product called uric acid, and that uric acid is excreted through excretory system through urine as a waste product. Intracellular nucleotides acts on purine nucleotides and convert them into nucleosides. They are put under strict metabolic control so as to ensure the various purine nucleotides are not depleting at their undergoing a depletion and to a below critical or threshold level. So these nucleotides are kept under strict metabolic control and they are degraded. A next enzyme called purine nucleoside phosphorylase that hydrolyzes nucleosides and liberate free purine base and ribose phosphate sugar. This sugar is further incorporated with inorganic phosphate and turned into ribose 1 phosphate. Notably, adenosine is not a substrate for purine nucleoside phosphorylase. Instead, adenosine is converted into inosine through a deamination, and that is mediated by adenosine deaminase enzyme. These pre-nucleobases are eventually converged into a pyrimidine ring or pyrimidine base called xanthine through various enzymes. Xanthine oxidase that converts inosine and turns it into xanthine, whereas guanine deaminase converts guanine into xanthine. And the xanthine thus produced is further oxidized to uric acid by xanthine oxidase. And this is an overview of purine degradation. The nucleotides called adenosine monophosphate, inosine monophosphate, xanthine monophosphate, and guanosine monophosphate are hydrolyzed to various respective nucleosides by an enzyme called nucleotides. Adenosine is converted into inosine through adenosine deaminase activity, and inosine is further converted into hypoxanthine, a free purine nucleobase. Similarly, xanthosine is converted into xanthine, guanosine is converted into guanine. And all these steps are mediated by purine nucleoside phosphorylase. The hydrolysis of nucleoside into pre-nucleobase, purine base, and a liberated pentose sugar is further incorporated with inorganic phosphate and liberated as ribose 1 phosphate. And through deamination, guanine is converted into xanthine and hypoxanthine is converted to xanthine. And xanthine is taken further through oxidation and converted into uric acid. And this is a structural depiction of purine degradation. Here it is adenosine monophosphate, inosine monophosphate, xanthine monophosphate, and guanosine monophosphate. They are hydrolyzed to our respective nucleosides through action of nucleotides, and these nucleosides are further hydrolyzed to have a free purine bases such as hypoxanthine, xanthine, and guanine. The enzyme that acts on nucleoside is purine nucleoside phosphorylase that hydrolyzes glycosidic bond and liberates free purine base and sugar, which is further incorporated with phosphate and liberated as ribose phosphate 1 and taken back to metabolic pathways. Hypoxanthine and guanine is further converted to xanthine, and xanthine is oxidized to uric acid, and this uric acid is excreted through excretory system. Accumulation of uric acid can have clinical manifestations. In case of enzymes involved in purine metabolism, such as hypoxanthine guanine phosphoribosyl transferase, the absence of this enzyme, HGPRT, leads to a clinical manifestation called Lesch-Nyhan syndrome and hyperuricemia. Similarly, an enzyme called phosphoribosyl pyrophosphate synthase or pyrophosphate kinase, defects in this enzyme leads to gouty arthritis. Defects in adenosine deaminase enzyme manifest severe compromised immune disorder. Similarly, enzymes involved in pyrimidine metabolism also give rise to defects of those enzymes, give rise to clinical conditions called orotic aciduria type 1, type 2, megaloblastic anemia. And that's all for this. Thank you and happy learning.